A very special evening to all of you, and welcome to the Nextopia Medical Lectures in the series of embryology lectures, the chapter 5, the fifth video which I am talking today will be the development of embryonic disc. Give me a second so that I settle down the audio and video, and then we'll start our hello. So in the series of lectures, this is the fifth one. The series is I'm talking about embryology, okay, human embryology. In the last class, we have discussed development of embryonic disc, and I hope you remember that we were talking about the embryonic disc, how the primitive streak and the notochordal plate begins to develop. There is some disturbance going on here. I'm really sorry for that. I'll settle it down. So, am I audible properly now? There's a lot of disturbance. Hope I'm audible now. Hello. 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 So this part of embryology is considered as slightly dry because it has got few unacceptable points, and these points tend to stay in our understanding with vague inputs, because once I'm talking about embryonic disc, I'll be talking three-dimensional kind of a picture, and which makes you to rather very difficult to visualize. So I told you before starting anatomy, embryology, histology, whatever it is, the second series of lectures after completing physiology, I already told you that unlike physiology, in anatomy, you need to develop a strong visualization. When I am talking about visualization, you need to draw certain diagrams, your own kind of diagrams. You need not be artistic in your diagrams, so that those diagrams will help you to understand where lies what, so that you come up with correct answers in the MCQs. Yeah. audio is settled, video is also good. Let us begin the discussion. So first, as our habit, you know that we will be first discussing, we'll first summarize what are the points that we will be discussing in the video today. We are discussing the further development of embryonic disc, in this, the first point that we will be discussing is, we'll talk about the cranial end of the primitive streak. You will have to understand these terminologies. Cranial end of the primitive streak primitive streak. You are calling it as a primitive knot. We'll discuss this first. After that, see, we are summarizing what we are discussing in the video. After that, what we'll read is, we'll read how the cells in the primitive knot undergo mitotic divisions, they multiply, they grow cranially into a rod-like structure called as notochordal plate. All right, notochord, or it's better to call it procaudal plate. Procaudal plate, or you can also call it notochordal process. Better term is procaudal. Small out projections, you are calling it as a processes, notochordal processes. After that, we'll discuss how the notochord suddenly disappears. First, notochord processes will, develop at a very rapid pace into a rod-like structure, and suddenly it disappears. It disappears, and the remnants of this notochordal process. So it's a rod-like structure which the cells disappear, only few cells remain throughout our life, which form the nucleus pulposus of your spine. You have heard that somebody coming with chronic back pain person coming with a long-term history. So we have got the vertebral body like this. All right, it has got a spine like this, posterior spine, and in between the two vertebrae we have got a space. You call it intervertebral space. Here you have got the intervertebral disc. All right, this is intervertebral disc. Intervertebral disc is, if you see from the top, this is from the side. If you see from the top, the intervertebral disc will be something like this, is it not? Sitting in between the two vertebrae. So the intervertebral disc in the center, in the central portion, there is a painless structure. I'm talking about painless because it is devoid of any pain fibers. All right, there is no, this is a sponge-like structure containing small amount of fluid in it, and this runs throughout this intervertebral disc. This is called as nucleus pulposus. Sometimes there is outpouching of the nucleus pulposus like this because of chronic bad habits of sitting or driving your vehicle, or people who strain their spinal cord a lot, they don't exercise, obese people. What will happen is this, there is herniation of the disc. Herniation of the disc. You call it herniation of the intervertebral disc. You also call this condition as a prolapse. Prolapse of the intervertebral disc. Prolapse of the intervertebral disc. Sometimes it's so painful. The nucleus pulposus is devoid of pain fibers, but once it prolapses, what happens is it blocks, it pressurizes the spinal nerves, and because of the pressure effect the person develops a lot of neurological disorders, and we'll have to cut it and remove it as a part of spine surgery. So this nucleus pulposus is derived from where? It is derived from the procaudal plate. And where is this procaudal plate derived from? From the primitive knot. This is what we are going to study today. We will also talk about one important point which we left in our last class, that is intraembryonic mesoderm. We'll see what is this intraembryonic mesoderm all about. We'll see how this intraembryonic mesoderm undergoes subdivisions, how it undergoes subdivisions into paraaxial mesoderm, into lateral plate mesoderm, and into somatopleuric layer, that is intermediate mesoderm. So we'll see how it divides into the paraaxial, the lateral plate, and the intermediate. We'll discuss what is this divisions of intraembryonic mesoderm in detail. We'll also discuss this intraembryonic mesoderm will have a cavity, we call it intraembryonic coelom, which is giving rise to the pericardium, the pericardial space. Coelom means space. Coelomic fluid is the fluid which is occupying that space. So how this intraembryonic fluid, the intraembryonic coelom is forming the pericardial cavity, the pleural cavity, and the peritoneal cavity, We'll discuss all these points. Next we'll discuss the leftover embryonic disc. How does it develop? The leftover embryonic disc, which was first flat, it has got a cranial end and caudal end now. So we can call it, it has got a head and tail now. And this head and tail undergo multiple folds, lateral folds, to form foregut, midgut, and hindgut. The endoderm especially. The endoderm undergoes a lot of foldings to form foregut, the midgut, and the hindgut. We'll discuss these points. We'll also discuss how the umbilical cord is formed. We'll also discuss how the foregut divides into the notochordal processes around the foregut will form the buccopharyngeal arch or the buccopharyngeal membrane. We'll also discuss how hindgut is limited by a cloacal membrane. And we'll also discuss in our today's class about the umbilical cord. So without wasting much time, let us start the discussion. So in the last class, I told you that in second or third week of development, there is a change of embryonic disc. The embryonic disc from circular to oval, then to pear shape, the embryonic disc is changing. And during the second week to third week, the second week marks the appearance of bilaminar embryonic disc, bilaminar embryonic disc, that is bi means two. So what are the two layers here? Epiblast and hypoblast. In my last class only I introduced these terms. Then I also told you that these will contain two cavities, and these two cavities you are calling it as a amniotic cavity and the yolk sac. Amnion and the yolk sac. So amniotic cavity and the yolk sac cavity, and these two are suspended by a stalk. Please remember that there is an axis formed. This axis it could be from head to tail. You are calling it as a cephalocaudal axis. There is one axis called as which goes from head to tail, cephalocaudal axis. Another axis you can ask is dorsoventral axis, from dorsal area to ventral area, dorsoventral axis. Dorsoventral area, that is dorsoventral axis. And you can also divide it into right and left axis. right to left axis. All right. Now, why I'm talking about this axis is the cells of the epiblast and the cells of the hyperblast. So they are actually moving along this axis. Movement of these cells along the axis is very, very unique, so that further growth and development of organ systems and various tissues occur. So please remember that the ectodermal cells, we have got ectodermal cells, is it not? Please remember that the ectodermal cells they are pluripotent. In my previous classes I told you what is pluripotent. All right, so pluripotent means they give rise to multiple different sets of stem cells. They give rise to multiple different sets of tissues or organ systems. So the pluripotent cells, the ectodermal cells, which are pluripotent in nature, they form, first is they are responsible for formation of definitive endoderm. They are responsible for formation of definitive ectoderm. They are responsible for formation of primordial germ cells. If you remember, I told you primordial germ cells, I told you these are the cells which are responsible for production of what? The spermatozoa and the ova. I told you in different spermatogenesis or oogenesis or gametogenesis class I told you. Now the ectodermal cell is also responsible for formation of a notochord. This notochord is actually developing into central nervous system in future. Then the pluripotent ectodermal cells also derive to form the mesoderm. So apart from mesoderm, ectoderm and endoderm, the two different points that you have to remember is they're responsible for formation of gametocytes, that is the precursors of gametocytes, that is the primordial germ cells, another is notochord. What happens is there is active migration of these cells. There is active migration means movement. The active movement of ectodermal cells and they're differentiating into ectoderm, endoderm. In between, what happens is when the cells are moving, what happens is a median groove is formed. So when the cells are moving means movement means new group of cells are added. So in between these cells, a median groove is formed. This groove you are calling it as a primitive streak. This groove you are calling it as a primitive streak. This groove you are calling it as a primitive streak. Now this primitive streak results in development of notochord. This primitive streak is responsible for development of the third germ layer, that is the intra-embryonic mesoderm. From the cells of the primitive streak, these two important structures take their origin. And after that, what happens is in the early part of the third week, a trilaminar, so second week I told you, what second week marks the bilaminar embryonic disc. Third week, by end of third week, what happens is a trilaminar disc is formed. When I say disc, I'm talking about embryonic disc, a trilaminar disc. So you can differentiate now from second to third week what are the important changes that are occurring. Now let us talk about a new term that you have learned today, that is the notochord and intra-embryonic mesoderm. We'll talk in detail. So what is notochord? Let us try to understand notochord now. Notochord is actually a midline structure. Please remember, notochord is a midline structure that develops where in between the head portion and the tail portion of the primitive streak and procaudal plate. All right, so it develops in between the caudal end of primitive streak So it develops between the cranial end of the primitive streak and the caudal end of the procaudal plate. The caudal end of the procaudal plate. The caudal end of the procaudal plate. During the development of notochord, there are multiple stages that are occurring. So what happens is, initially the cranial end of the primitive streak becomes thickened. The thickened part of the streak, you are calling it as a primitive knot. Just now I told you that is Hensen's node. It is also called as Hensen's node. So that procaudal plate or primitive knot, the primitive streak, so for example, if these are the cells of the primitive streak, tall columnar cells of the primitive streak, the median portion in between the ectoderm and the endoderm, which form the primitive streak. What happens at one end of this primitive streak? Say suppose the cranial end of the primitive streak, there is formation of suddenly differentiation into multiple cells like this. All right, small cuboidal cells like this. So this is looking like a knot, so we are calling it as a primitive knot, also called as Hensen's node. All right, so this layer from here to here, from here to here, it is the developing primitive streak. This is the primitive streak. Hope you are able to know it. Just beneath this, we have got another layer called as procaudal plate. So the caudal, so notochord develops in between the cranial end of the primitive streak and the caudal end of the procaudal plate. So this is the procaudal plate. If I try to draw this in a developing embryo, it looks like this, something like this. I'll draw it here separately. So it looks something like this. So this is the developing embryo. It has got a stalk with which it is attached to the endometrium. All right. So somewhere here, like this. Process is formed. You are calling it as a primitive streak. The cranial end of the primitive streak develops a small knot-like structure. What are you calling it as? A primitive knot. So this is the primitive streak. At the caudal end of this primitive streak develops what is known as a membrane. We are calling it as a cloacal membrane. Now this is a new term. Please note it down. Cloacal membrane. Cloaca means the hind part, that is in future, which is forming the anus and all. All right. Now what happens is in this primitive streak, in this primitive knot, what happens is there is a small pit. I'll draw it with which color? Green color. Yeah. So there is a small pit developing in this region of primitive streak somewhere here. This you are calling it as a primitive pit. Or a primitive groove, also called as a blastopore. Its another name is blastopore. Its another name is blastopore. what's happening to this blastopore? In this blastopore area, the notochordal processes start, and thus there is growth of the notochordal process. The cells of the primitive knot, they are growing like this in between the cranial end of the primitive streak to the caudal end of the procaudal plate. All right, so these are the cells growing like this. In these cells, the blastopore is growing like this. All right, you follow the green color now. Follow the green color. So in this direction. All right, so this is all the notochordal process. We are calling it as a notochordal plate with a notochordal canal. So this entire this structure you are calling it as notochordal process or notochordal plate. And in between the green color area, you are calling it as a notochordal canal. Notochordal canal. All right. A small depression, I repeat, a small depression appears in the primitive knot. You call it primitive pit. The cells of the primitive knot multiply and they pass cranially in the midline in between the ectoderm and endoderm. So in between the black colored layer, that is primitive streak ectoderm, and the blue colored layer, that is the endoderm, in between these two, from the cranial end of the primitive streak to the caudal end of the procaudal plate, the pink color cells which are multiplying and multiplying, these cells are forming what? The notochordal process. Now the notochordal process, the cells they are actually solid, completely solid, means there is no cavity or canal in it. So this is head process of the notochordal process. Now what happens is a solid rod like structure. Notochord means, the name of why are you calling it notochord is, in Latin it is similar to a solid rod like structure. A solid rod like structure. So what happens? A small pit develops in the primitive knot which enlarges and forms the notochordal canal, the green colored structure in this diagram. Notochordal canal. The cells forming the floor of notochordal canal become the cells of the endoderm. The cells forming the floor of the notochordal canal, they become the cells of the endoderm. The floor of the notochordal process canal begins to break down, and at first what happens is small openings are found, but gradually whole of the canal communicates with the yolk sac below. So if you remember my last class, I told you where is this yolk sac. It is just beneath the blue color lining, that is the endoderm. So what happens, the cells below cells are slowly, they are getting dissolved, and these cells, they are opening up the channel into the yolk sac, and they are forming a communication. Thus at this stage what happens, so here on the top you have got amniotic cavity here. I'll draw it with black color. Amniotic cavity here. All right, and here you have got the yolk sac. If the cells in the floor of the canal, they are breaking down here, what's happening now? There is a communication formed between the amniotic cavity and the yolk sac cavity. Now the two cavities are communicating with each other. Gradually what happens is the cells, gradually what happens is the walls of the canal become flattened, so that instead of rounded canal, we have got a flat plate-like structure. Now you are calling this plate-like structure as a notochordal plate. So because of modification of the shape, you are calling it as a notochordal plate. Now what happens to this flattening cells? Soon what happens is the flat cells again become round or curved, and they take the shape of a tube. All right, initially the cells are flat. Now the flat cells will round from the borders, and they go and meet in the midline and to form a tube. Now you are calling it as a notochordal tube. So from the notochordal process to the notochordal plate, from the notochordal plate to the notochordal tube. This is the chronology of formation of the notochord. As the embryo enlarges, the notochord also elongates, and it is lying in the midline. it is responsible for formation of the vertebral column or the spinal, your spine. All right, that is responsible for formation of your vertebral column. And what gives rise to the parts of the spinal column, we'll try to understand. All right, so the primitive streak was the prime organizer which started the development of notochord, is it not? And it also started the development of intraembryonic mesoderm. Hence, the primitive streak is very, very important, especially in the second week of fertilization, immediately following second week of fertilization, beginning of the second week of fertilization, all this process takes place. They are very, very important for formation of the central nervous system. Please remember this part. Now, how is this? I told you that the notochord becomes plate, the plate becomes tube. How is this happening? Let us try to understand. So initially what happens is the neural tube gives rise to, we should know that this notochordal plate, it is forming notochordal tube, and this tube is responsible for formation of what? The spinal cord in the future. And it is also responsible for the cranial end will form the brain, will differentiate into the brain. So it is such an important structure, and it is occurring the second week to third week of the fertilization of the intrauterine life. You can imagine how early these structures are formed. And the neural tube, soon it is divisible into two parts, the cranial part and the caudal part. The cranial part is brain, which enlarges to form the brain, and the caudal part is the spinal cord, which elongates to form the spinal cord and gives rise to branches called as spinal nerves. In the case of intraembryonic mesoderm, now very, very interesting point is intraembryonic mesoderm. Now the most important aspect is intraembryonic mesoderm. What happens in intraembryonic mesoderm is the cells of the intraembryonic mesoderm that are formed in the primitive streak, that are formed because of proliferation of primitive streak that separates the ectoderm and endoderm. There are few regions wherein there is communication between these two areas. I told you that the procaudal plate, there is one structure called as procaudal plate in the endoderm, is it not? So this procaudal plate is responsible for development of the future oral cavity. You call it buccopharyngeal membrane. Buccopharyngeal membrane. Buccopharyngeal membrane, which is forming the future oral cavity, mouth. All right, this is the first aspect. Next, the tail end, I told you, develops into the cloacal membrane. This cloacal membrane is responsible for future anal opening. Anus. All right, these are the parts of the intraembryonic mesoderm, from the cranial part to the caudal part, from the cranial most part to the caudal part. So the cranial to the procaudal plate, the mesoderm of the two sides meets in the midline, and this midline union of the mesoderm forms what is known as extraembryonic mesoderm. Please remember the intraembryonic mesoderm. Now it has got three parts. Intraembryonic mesoderm, the mesoderm on the either side of the notochord is one part. The mesoderm which forms the thin layer is the second part. The mesoderm which is having the longitudinal strip, it is the third part. So the mesoderm which is on the either side of the notochord, if you see this diagram, the mesoderm which is on the either side of the notochord, mesoderm which is on the, so either side of here, either side of the notochord, the mesoderm, intraembryonic mesoderm on the either side of the notochord, it forms paraaxial mesoderm. The mesoderm which is forming the thin layer and which is more laterally to this aspect, I'll draw it with green color. Much more lateral, this area. Much more lateral, this area. This is forming what is known as the because it is lateral, you are calling it as a lateral plate mesoderm. Lateral plate mesoderm. And in between these two, that is the paraxial and lateral plate mesoderm. So I'll draw it with a highlighter like this. In this area, the pink color highlighters, they form what is known as intermediate mesoderm. What are the derivatives of this? We'll discuss. Intermediate mesoderm. These are all the important divisions of the intraembryonic mesoderm. So first, the paraaxial mesoderm. The paraaxial mesoderm, the cells are linear, homogeneously arranged. The mesoderm gets small segments in this. Small segments. These small segments, they develop into somites and somatomeres. So this paraaxial mesoderm divides into somites and somatomeres. There are 7 rounded structures. You are calling it as a somatomeres. Somatomeres are the cells which lie in the region of the head. All right, there are 7 of these. Somites are cubical and distinctly segmented areas, and somites are usually formed in the cranial aspect, and they progress caudally from cranium to caudally. There are 44 pairs of somites. There are 7 somatomeres and 44 pairs of somites. I'll write it again. So the paraaxial mesoderm divides into somites, somatomeres, and somites. There are 7 regions of somatomeres. All right, 7 regions just lateral to the notochord. They form what? They form the muscles. In the future, they develop into muscles. Whereas somites are cubical structures. They are well organized, well oriented, small cubical structures. They are 44 in number. They are 44 pairs. All right. Here 4 are occipital, 8 are cervical, 12 are thoracic, 5 lumbar, 5 sacral, and 8 to 10 are caudal. Now you can imagine they form the vertebral somites. From these somites develop that region of the whole body. The somite forms an axial skeleton. All right, each of these somite forms an axial skeleton. Please remember that somatomeres are responsible for formation of muscles of the head and jaw only. Somatomeres, they form the muscles of head and jaw only, whereas muscles of body are actually divided from these 44 pairs of somites. Each somite gives rise to axial skeleton. It means bone. It gives rise to axial skeleton. It gives rise to a skeletal muscle. And it gives rise to skin. So say suppose one somite gets infected in herpes zoster infection. It will affect the same skin supplied by this somite. It will affect the same muscle. It will affect the same axial skeleton. Similarly, if a person develops myocardial infarction or a heart attack, he develops pain in the chest. Problem is in the coronary artery, but he develops pain in the center of the chest, and the skin over the chest is actually painful for him. The reason is the coronary artery which is formed, that somite, the somite that is responsible for the myocardium development, the somite which is responsible for the development of the skin in the center of the chest are same. Hence the pain is observed here, and on the lateral aspect of your left hand, up to the inner aspect of the left side of the jaw. So this is the distribution of pain in the heart attack. It is because of this somite. Each somite forms axial skeleton, the skeletal muscle, and part of the skin. Please remember that somatomeres are not confined to the region of somite. So somatomeres are confined to the head region. From head to toe, somites will come and they take over. So we'll talk about further about somites and somatomeres in other chapters of development of muscle and development of skin and all axial skeleton. We'll talk about it later on. My internet is showing some problems. Give me a second. I think I'm back. So we were discussing about the paraxial mesoderm. This is all about the discussion. This is the discussion of the paraxial mesoderm. The next part of the mesoderm is the lateral plate mesoderm. The lateral plate mesoderm, while the paraxial mesoderm is undergoing segmentation into somites and somatomeres, what happens to lateral plate mesoderm? There are small cavities formed in the lateral plate mesoderm. Small cavities are formed in the lateral plate mesoderm. These cavities will close together, they come close together and form a large cavity. So I told you that this is the notochord. Say suppose this is the developing notochord. All right. What is in the head end portion of the notochord? We have got the primitive knot. What is in the tail end of the notochord? We have got the cloacal membrane. All right. So somewhere here we have formation of the paraaxial mesoderm. Para means beside, axis means center. So just beside the notochord, the development of mesoderm into somites and somatomeres is occurring. We are calling it as a paraaxial mesoderm. Just beside this, we have got lateral plate mesoderm. So this area. In here what happens is small cavities are formed first initially. Later on these cavities will coalesce. That is they mix with each other and form a large cavity. They merge with each other. They merge with each other to form a large cavity. Alright, they merge with each other to form a large cavity. With the formation of this cavity, you are calling it as an intraembryonic coelom. It's a very important terminology. Please remember. Intraembryonic cavity or intraembryonic coelom. This intraembryonic coelom splits into the somatopleural and parietal layer, and they come in contact with the ectoderm. So it splits into the somatopleuric or the parietal and splanchnopluric or the visceral layer. Parietal means outside, visceral means the organ system. So it splits into two halves. So this part and this part, say suppose it has got two parts, is it not? So this part becomes the parietal and this part becomes the visceral. I'm just correlating it, all right, just to help you to understand of this cavity. So the parietal comes in contact with the ectoderm outside, whereas the visceral will come in contact with the endoderm. It comes in contact with the endoderm layer. These layers are already formed. It comes in contact with the endoderm. So what is the function of this intra-embryonic coelom? This intra-embryonic coelom is responsible for formation of the important cavities like pericardial cavity around the heart, peripleural cavity or simply pleural cavity, and the peritoneal cavity, that is the abdomen. That is the abdomen. For time being, please remember that the pericardial cavity is formed from the intraembryonic coelom that lies in the midline of the procordia. So in the midline, it is close to the cardiogenic area. It forms the pericardial cavity. Close to the pleural area, it forms the developing lungs. It forms the pleural cavity, and developing intestines, it forms the peritoneal cavity. All right. The mesoderm does not split along the intraembryonic coelom. If the intraembryonic coelom has not extended up the mesoderm, it does not split. And the area of mesoderm which does not split, sometimes I told you that small cavities are joining to form the large cavity, but sometimes if these areas don't split, they form what is known as septum transversum. Now what is the septum transversum? We'll learn it in future, but please remember this term, septum transversum. It is the area of the mesoderm which has not split, so it forms the septum transversum. All right. So we have discussed what? We have discussed clearly, we have discussed important points like the development of paraxial and lateral plate mesoderm. What happens to intermediate mesoderm? Intermediate mesoderm derives the intermediate mesoderm of the urinary and the genital system. Intermediate mesoderm so it helps in giving rise to the mesoderm of the urinary tract and the genital tract. See, the terms are very, it doesn't give rise to genital tract, it gives rise to mesoderm of the urogenital system. It doesn't give rise to urogenital system, it gives rise to only mesodermal component of the urogenital system. I'll tell you what is that in the chapter of urogenital embryology. But for now, please remember these basics are very, very important. All right, these basics are very, very important because from bilaminar disc it has become trilaminar. So now let us discuss a very, very important aspect, that is the folding of the embryo. All right, let us discuss folding of the embryo. It's a very, very important concept to understand. So we have got the head fold, the tail fold, the midgut fold, all right, the foregut fold. So how the amniotic cavity and the yolk sac is helping in folding, let us try to understand. So what is embryonic folding or folding of the embryo? Let us try to concentrate now. So if you see this folding of the embryo, the folding of the embryo, it is a progressive process. What happens is there is an increase in the size of the embryonic disc due to rapid growth and addition of multiple cells. Because of this increased size of the embryonic disc, the central portion of the embryonic disc undergoes rapid growth, very, very rapid growth. This causes conversion of a flat pear-shaped embryonic disc into a cylindrical embryo. All right. So first what happens? A flat shaped embryo has now become a cylindrical embryo. You can imagine the shape, cylindrical. Now this cylindrical, I'll draw a cylinder like this. So now this cylinder has got a head end and a tail end. It has got an axis, head end and a tail end. However, These are very close to each other. They are not away from each other. But with increasing length of the disc, what happens is it causes a bulge into the amniotic cavity. And this bulge into the amniotic cavity is where the folding of the embryo is occurring. How? So what happens is the edges of the embryonic disc, they become folded on itself first in the median plane and the transverse plane. And this undergoes a folding like this. You call it a head fold. This undergoes a folding like this because of growth of cell. You're calling it a tail fold. So we have got a head fold and a tail fold. Head fold and a tail fold. All right. Now with the formation of head fold and a tail fold, there are three separate folds actually formed. There is also a lateral fold forming, transverse plane. All right, lateral fold. All right. So this is in the anteroposterior, the craniocaudal axis, but laterally also a fold is formed. I cannot show you in this diagram. I can simply write there is third fold called as lateral fold. Now these three folds, the head fold, the tail fold, and the lateral fold. Now these three folds and few small part of the cavity, that is yolk sac, they become enclosed within the embryo and forms a tube. A tube which is lined by the endoderm is formed first. A tube which is lined by the endoderm is formed first. All right. So if say suppose this is the plate, now plate is growing like this. It is forming the tube. It is forming the folds, and the lower portion of the fold is filled up by the endoderm, and this is lined inside by the endoderm. This we are calling it as a primitive gut. So you understand how the primitive gut is formed. Now this primitive gut, if you talk about the cylinder which I was talking, it has got a head and a tail, is it not? Now it is undergoing folds like this, folds. So the head portion, say suppose this is the head portion, this is the tail portion. The primitive gut in the head portion forms the foregut. In the middle portion it forms the midgut. In the tail portion it forms the hindgut. Gut means the intestinal precursors. So we have got formation of the midgut, foregut, and the hindgut, and there is a communication with the yolk sac. The yolk sac communication is becoming narrower, and future it will there is a small definitive yolk sac. As a result, what happens is changes in the yolk sac, they become narrower, and in this area the embryo is attached with the help of a duct, with the help of a stalk. All right. So I'll draw it like this. So we have got a embryonic disc. We had, if you remember, amniotic cavity and a yolk sac cavity, something like this. So this was amniotic cavity, this was yolk sac, and this is the blue color one is the embryonic disc. What's happening to the embryonic disc? It is growing like this. It is growing like this. Now it is slowly engulfing the yolk sac. It is forming the gut, foregut, midgut, and hindgut. So this yolk sac, small amount of yolk sac, somewhere here, it is with the communication with the gut like this. This communication. This we are calling it as a vitello-intestinal duct. Small communication that is formed through the yolk sac to the stalk, and this stalk is communicated where? To the uterus at the level of placental form, placenta. So it is forming a definitive yolk sac, and here what is formed is the umbilicus or the umbilical vesicle. Here forms the umbilical vesicle, and the duct inside you are calling it as a vitello-intestinal duct. This vitello-intestinal duct is also called as vitelline duct, also called as yolk stalk, also called as omphalomesenteric duct. Remember the other names. It is also called as vitelline duct, or it is also called as omphalomesenteric duct. Omphalo means umbilicus, mesentron means midgut, mesenteric, omphalomesenteric duct, or it is also called as yolk stalk. All right, if you remember. The lateral folds, which are transverse plane and horizontal plane, they actually enclose all around by the ectoderm, except in the region of the vital intestinal duct. Here it is the umbilical cord formed. What are the structures of the cord? We'll discuss subsequently. We'll discuss what are the structures of the umbilical cord. But if you remember, if you try to understand, this is how in the embryo, the umbilical communication from the embryo to the stalk, through the stalk to the placenta is slowly it is developing. All right. The connecting link between the embryo and the mother is the connecting stalk. Through this stalk, we have got the vital intestinal duct, and it is communicating to the placenta. It is communicating to the, as the embryo grows, what happens? As the embryo grows, the area of attachment of this stalk becomes smaller. You can imagine the embryo grows and becomes a fetus. The area of attachment becomes smaller and thinner and well organized. So if you can see, what are the structures that are present in this stalk? There are blood vessels in this stalk, and these blood vessels are very, very important because they are carrying nutrition from the mother to the fetus, mother to the growing embryo, and it is present on the ventral aspect of the embryo. So if you can see this embryonic disc, all right, say suppose here somewhere the umbilicus is growing like this, all right, to the midgut. Here the dorsal structure that is formed is notochord. Dorsal structure that is formed is notochord. And in the ventral aspect, what is the important union that is forming? That is the vitalo-intestinal duct. Now this vitalo-intestinal, what are the structures that are present here? What are the structures that are developed in the umbilical cord? Let us try to understand. There are sets of blood vessels. If you take, so if this is the growing fetus embryo, all right, so suppose this is the tail of the embryo like this, and if this is the placenta, if this is the uterus, and in the uterus we have got a placenta like this. So this is the communication I'm talking about. So this is the communication which we are talking about. All right, so this is the growing fetus. All right, if you can see development of the notochord in this area, the dorsal aspect in this area. So I'm just imagine an imaginable diagram I'm just drawing. So if you take a cross section of this area, if you take a cross section of this umbilical cord, you will find two sets of arteries and two sets of veins. So if you take a cross section of the umbilical cord, you will find two arteries and two veins. We have got right artery and right vein, left artery and left vein. All right, so we have got right artery, left artery, right vein and left vein, right vein and left vein. At first what happens, there are two arteries and two veins, but later what happens, the right vein disappears, the right vein disappears, and only the left vein is left. All right, if you can understand, the left is left. So the right vein disappears. So this is the cross section of the umbilical cord. The right vein disappears. The left vein is left. Whereas arteries are, so a mature umbilical cord is something like this. We have got two arteries, cross section, all right, and one vein, only the left vein. So we have got left umbilical vein, we have got the left umbilical artery, and then we have got the right umbilical artery. These are the contents of the umbilical cord. And then in between the cord, we have got a gel-like structure which gives the strength, support to the cord. This you are calling it as a Wharton's jelly. This you are calling it as a Wharton's jelly. gel-like connective tissue. This is connective tissue. So these are the contents of the umbilical cord. We have got the vitalointestinal duct. Somewhere here we have got the vitalointestinal duct. All right. So this is the vitalointestinal duct. Vitalointestinal duct. Remnants of yolk sac. The Wharton's jelly. What is the Wharton's jelly? It is nothing but the mesoderm. It is the extra embryonic mesoderm. It is the extra embryonic mesoderm which protects the blood vessels in the umbilical cord. You have got the blood vessels which connect to the placenta. A small part of the extra embryonic mesoderm is also present. So this tube, it is surrounded by the amniotic membrane. Amniotic membrane. So this is the structure of the umbilical cord. At the time of birth, what happens? The umbilical cord is about 50 centimeters in length. 50 centimeters in length and about 2 centimeters in diameter. So it undergoes torsion with fetal movement, and at the time of birth it has to be cut safely so that the baby will not develop any abnormality related to the cutting of the cord. The baby does not develop any abnormality. So this is what happens. This is what is expected immediately following delivery. You will have to follow the safe practice of delivery. All right. So this is all about the folding of the embryo, folding of the embryo and the formation of the umbilical cord. So head fold, tail fold and lateral folds. If you remember, foregut, midgut and hindgut. In the head area we have got pericardiac cavity formed. Because of the division of the intraembryonic mesoderm, we develop pericardiac cavity, and this is in the cardiogenic area. Future heart is going to form, and the yolk sac is constricted into a small canal, and it is communicating with the mother in the form of vitalointestinal duct. So this is what you'll have to remember. If you have any doubts in this class, please feel free to ask me. This is the conclusion of today's lecture. All right. In the next class, we shall be discussing about the placenta. We'll discuss about the amnion, the chorion and all. So we'll talk about all those in the next class. Thank you very much for patient listening.